Okay, so I wanted to go ahead and give you some feedback here on this lipid transport worksheet. Of course, we're thinking here in chapter 10, 11 material, really chapter 10 because we're thinking about lipid transport. And so in that worksheet, you've been essentially asked to fill out a sequence of events according to these boxes here, and we've done this in worksheets previously. In which the very first thing that happens is this, and that is that the intracellular cholesterol levels drop. And then what is that going to do intracellularly that will then allow for those cells to either make cholesterol or pull in cholesterol from the bloodstream? Okay, and in that latter case, we're talking about the production of LDL receptors. And when that occurs, then that can actually cause LDL levels to be decreased in the blood, and we'll get to that in our what would be called step 2 of our worksheet, because it asks us to do some reverse engineering there. So let's think about this though. Intracellular cholesterol levels drop. Okay, that's what is the initiator event here. And what that's going to do is it's going to allow oxysterol. So I'm just going to put this here and then I'll refer over to our figure here. Oxysterols, okay, to release. I'm running out of room here. Let me release. And it would be NSIG and SCAP. Okay, so that's the very first thing that's going to happen. So if we come over here and we look at this, let me just highlight here for us oxysterol and sterols. Those are these little things that are pointed at. Apparently they're these little ovals, okay, either pink or orange ovals. Oxysterols and sterols are essentially products or byproducts of a cholesterol synthesis pathway. If cholesterol levels are high intracellularly, then the oxysterol concentration is going to be high. And when they're high, those are going to be bound to these two proteins, NSIG and SCAP. Right? Now, when we look at SCAP, that SCAP actually stands for SREBP, which is the S part, cleavage activating protein. Okay, so that's what that stands for, where SREBP stands for sterol regulatory element binding protein. Okay, so lots of terms there. Okay, I prefer SCAP and SREBP, but those are what they stand for. Okay, but that's the very first thing that's going to happen. Okay, now what's that going to do in terms of affecting cholesterol synthesis pathways and cholesterol intake into the cell is what we need to talk about next. Okay, so that's step 1, though, oxysterol release NSIG and SCAP. Okay, now the next part is this. Okay, and that would be, let's look at this. So we move over here, and if you think about it this way, we could even put this. They don't do this in this figure, but they could put oxysterol coming off, okay, of those things. And that then allows for this ubiquination, okay, of NSIG. Okay, so we would say this, NSIG is ubiquinated. Ubiquitinated. Okay. Okay, and that then marks it for destruction, okay, and we could just say degraded. We're not going to talk about how that happens, certainly degradation, but when you have what would be considered polyubiquination of a protein, okay, that can oftentimes mark that protein for degradation. Right, and so NSIG then is removed. Now, what that then allows to happen, okay, is that then these secretory proteins, okay, that were originally, they're kind of shown here, but they are shown here now. But that allows for these secretory proteins to bind to SCAP. Okay, so, and I'll just put, because this is a big one, it's a mouthful, secretory proteins bind S-cap and S-cap slash SREBP they move from ER to the Golgi. Okay, and that's what essentially, you don't really see it here, okay, but they're in the ER membrane here, okay, and then right when you have this ubiquitination, then the secretory proteins escort, that's the term here, escort S-cap and SREBP to the Golgi. They don't show that, but that's what happens. Okay, now in the Golgi, then let's get out another color here. Then we have then these proteases that SREBP will be exposed to. Okay, and those proteases are going to cut right here and right here. Right, so protease cleavage of SREBP. Okay, and that then releasing, I guess I should say this, releases, and I'll just put RE, regulatory element or regulatory domain or protein. Okay, that would be what happens here. Okay, now that regulatory domain, okay, so we see that it's kind of, you can kind of see it highlighted here, but this is the regulatory domain. That's going to then diffuse to the nucleus. Diffuse, oh wait, let's say RE. Diffuses to nucleus. Right? And then, so that's what happens here, that's this step here, and then finally, once it's in the nucleus, okay, the regulatory element, okay, induces genetic expression of, and it would be a host of genes, but it would be, whoa, sorry about that, it'd be a host of genes, and so we'll just say cholesterol metabolism genes. Okay, and so it says here, that's essentially what we're looking at here, transcription of lipid synthesizing enzymes stimulated. Okay, this would include, and this is important, LDL receptors. Okay, so remember, all of this happens because there's a drop in cholesterol, so then the cell needs cholesterol. Cholesterol is actually an important element of the life of the cell. It's a very important component of membranes, okay, depending on what cell you're talking about. It's also a secretory protein, in secretory glands, it can be used to make hormones. But so there's just lots of reasons that a cell needs to make cholesterol. So if those drop, then there's going to be this right whole sequence event. The oxysterol is going to drop, it's going to release N-sig and S-cap, and then N-sig is going to be ubiquitinated and degraded, and then S-cap and SREBP will then move to the Golgi. The SREBP will be cleaved by these proteases, the regulatory element will then be released, it will diffuse to the nucleus, and then that will bind at promoter elements of upstream of several different types of genes that are involved in cholesterol synthesis, including, okay, and important for our next part of our discussion, LDL receptors. Okay, all right, so that's what happens when we look at dropping of cholesterol levels. Okay, now as we then look at this, okay, the next question is this. I think that's it for that part. Yeah, that's that for that part. But it says in step 2, it says once you have done the above, let's do some reverse engineering and answer the following question. When a person has a genetic predisposition to high LDL content, how can a statin drug help? Okay, well, statins okay, are going to bind to one of the key regulatory elements, okay, or not regulatory elements, I'm sorry, but key regulatory enzymes within the cholesterol synthesis pathway. Okay, so cholesterol synthesis is quite complex, okay, but statins can actually inhibit cholesterol synthesis within a cell. Okay, now why would that be important? Okay, and now statin drugs, just so you know, these are drugs like Lipitor, things that you might see advertised during like a football game, or you're watching a TV show or something like that, and they're used for people that have high cholesterol, and that's a fairly significant portion of our population. And so these drugs then inhibit cholesterol synthesis within a cell. Okay, now why would that be important? Well, if you inhibit cholesterol synthesis within cells, in particular within the liver, then you're going to essentially activate this entire process that we just talked about, all of that, okay? And that will lead to then the production of these LDL receptors that will then be put onto the membranes of cells, in particular the liver. LDL will then be taken out of the bloodstream, and that will then reduce LDL concentrations within the bloodstream. Okay, and that will hopefully then stave off potential cardiovascular diseases that can result from high circulating LDL in the blood. All right, now that's it that I'm going to cover in terms of this recording. You can look at the rest of the worksheet. The rest of the worksheet is a crossword puzzle. Have fun with that. I'm not going to go over the results of that. Nice thing about crossword puzzles is that you typically know that you've gotten it right by the time you're done with it. Okay, so look at that. It can actually be helpful, just in kind of a fun way of just going over the definitions that we're talking about, specifically in chapter 10. Okay, all right, so take care, everyone, and if you have any other questions regarding this, please let me know.